morning. I'm truly honored for this award, and so thanks to the organizing committee, and a special thanks also to the Buiatti family. In my thesis work, I studied what happens when cells interact with specific materials, and in particular, I focus in what happens at the nanoscale using advanced nanoscopy. So the idea is what happens when cells interact with a specific substrate. This interaction is quite well known when we speak about macroscopic function of the cells, and I think that it's fascinating if we consider stem cells, because the stem cells interacting with the material and depending on the physiochemical properties of the material can differentiate in different lineage. And this is quite well known in a macroscopic way, but what happens at the basis and what happens inside the cells, so the mechanism behind this interaction, are unknown. So the idea of my thesis is to change scale of observation and to go inside the cells. And I developed a protocol to study what happens inside the cell's nucleo, the chromatin organization, using super resolution microscopy when the cells interact with 3D printed scaffold. So just to introduce some biological concepts, the chromatin is the condensed form of DNA within the cell's nucleus, and in this field is quite important because it controls the gene expression, while the stem cells are cells that are not differentiated and can specialize in different cell types, so either osteogenic cells or neuronal cells. And this could happen when interact with a substrate that I call scaffold. So my thesis is divided in 3 different phases. In the first one, I use super resolution imaging to characterize chromatin organization. In the second one, I fabricate and characterize 3D printed scaffold. And finally, I achieve super resolution imaging of the cells grow on the fabricate 3D scaffold. Inside the chromatin, I focus on 2 different histone modification. We can say 2 different proteins, which are H3K4-trimethyl and H3K9-trimethyl. And this because they are generally associated respectively with active genomic regions and inactive genomic regions. But the problem is that the chromatin forms aggregates, and the dimension of this chromatin cluster is about 10-100 nanometers. So the problem is that the traditional microscope that use visible light are limited with the diffraction limit, which is about 200-300 nanometers. So to observe the chromatin cluster, we have to go beyond the diffraction limit and use super resolution imaging. Within the super resolution imaging techniques, I use a technique which is single molecule localization microscopy. And with the single molecule localization microscopy, we can break this diffraction limit using photoswitchable probes and divided in time the emission from different molecules. So frame by frame, I can see the emission of few molecules, and then frame by frame, I can processing them, determine the coordinates of each molecules, and then aggregate the localization together. And this is possible using the triplet state of the molecular energy levels. So we can see at the right in a conventional imaging, conventional image of the chromatin, while on the left, in the right, sorry, a super resolution image of chromatin. While here we can see the 2 different proteins that I looked. So at the left the active regions of chromatin, while at the right here the inactive regions of the chromatin. And thanks to this type of techniques, so single molecule localization microscopy, we have, yes, an image, but we have also the coordinates of each molecules of my sample. And this is quite important because it allows us to implement different quantitative methods. Here I started from the localization, and then I divided in aggregates, so in chromatin clusters, depending on the density of the molecules. And here, different colors represent different nerve clusters. Then we can quantify the properties of the clusters, like the area and the number of localization per cluster, and we can see how the inactive active regions have different properties of inactive regions. And here we can observe also a double population for inactive regions. Important to say that this property will not have been possible to observe using other techniques of microscopy or other techniques of the chemistry side. Before I use the scaffold to differentiate the cells, I differentiated the self with biochemical differentiation. So I start with mouse embryonic stem cells, and then I differentiated them into neuronal progenitor cells. And this phase was necessary to be sure that our protocols could be used to see difference between different population of cells. So we can see that we have statistical differences between stem cells and the neuronal progenitor cells. Okay, in the second phase, I fabricate and characterize the 3D printing scaffold. I use 3D printer, which is an extrusion-based technology that enables the realization of our structure filament by filament and layer by layer, extruding a polymer through the application of a control pressure and in a moving cartridge. And in this phase, I use a polycaprolactone, which is a biocompatible polymer, and it's useful because it's biocompatible, but there are studies where polycaprolactone can influence and differentiate the staminal cell. But before starting with the fabrication of scaffold, it's important to understand the material rheology and then how the printing parameters, like the speed of the cartridge and the pressure of the extrusion, influence the diameter of the filament printed. So we modelized and demonstrate our model for the diameter of the filament in function of the pressure and the speed. And then we characterize, we fabricate 3 different types of scaffold: flat scaffold, parallel scaffold, and perpendicular scaffold. And then we characterize them using using SEM and using profilometry measurements. And finally, we cultured cells on the 3 types of scaffold, and we demonstrate that it's possible to produce images of the cells cultured in the scaffold using an inverted microscope. And we also observe that the cells appear to follow the geometry of the scaffold, so they go straight in parallel scaffold and they settle into the pores in perpendicular scaffold. But the most important results is that we are able to produce a super resolution image of cells that grown on our 3D scaffold. And this image is quite innovative in the field because there are studies that are studying the chromatin, but in the cells cultured in traditional glass substrate, while other studies are studying cells, but only the macroscopic properties of cells. So in my thesis, we change the observation of this phenomena, and we demonstrate that it's possible to study what happens inside the cell's nucleus when the cells interact with a specific substrate. So to conclude, I characterized the chromatin using super resolution images. I demonstrate that there are quantitative differences between different types of cells. I fabricate and characterize 3D printing scaffold, and then I demonstrate that with this protocol is possible to go inside the cells to study what happened during the interaction. So thank you. Thanks to my supervisor, Professoressa Francesca Cella Tonacchi and Professor Dario Pesignano, and to the other people that helped me during my thesis work.
Any questions?
You said the fluorescence.
Yeah.
Okay. But there is the possibility to use phosphorescence or that type of methodology to get images. Because you can use phosphorescence in
Yeah.
You can do a parallel.
I use fluorescence because there are a lot of techniques that try to go in the super resolution way, and the single molecule localization was fluorescence because the time of fluorescence is about 10⁹ nanosecond, and the most important thing is that we can divide the fluorescence emission of the molecules in time. But with phosphorescence, I'm not sure if could be possible to do this photo bleaching, and so I'm not sure if we can divide it also the time of the emission of molecules. But there are other methods that can do that. This working on the illumination of the sample. So there are methods that engineer how I can illuminate my sample, or there are other methods that don't use light but use electron. But with the chromatin, there are more problems. So. For the chromatin, also in literature, I think that single molecule localization microscopy is the best, but for sure there are other type of techniques that could be useful and also to integrate in the same measurement. Okay. Yes.
The one is if with your method you were able to make.
So that everybody
stem cell become a neural cell, okay?
Yeah.
You can have two kinds of neural cell, excitatory cell and inhibitory cell.
Yeah.
Did you have the possibility with your imaging to distinguish between this kind of cell? Because this is important.
Yeah, this is important. For the moment, I differentiate in neuronal progenitor cells, so in the phase before. I think that, I don't try this, but could be done, but I think that also to look the chromatin for dividing the two population of neurons could be useful. Also mark other proteins in fluorescence that could be present in a lineage and not in the other. So maybe, I don't know, but I think that could be done in parallel, chromatin measurements to see if the clusters are different, but of sure also another protein level to I will find the protein that I can observe in one type and not in another to see if the population are different. But thanks for your question.
Thank you for this amazing work. I have two questions.
Okay.
One given by my ignorance of these systems is very simple. How much time do you take to do these images? Because I imagine that the cell then transforms in time. So what is the, let's say, the temporary resolution of?
But temporary resolution of one image or of when I see the.
How long do you need to observe image the cell to get these images? Then I guess you have to reconstruct, etc.
Yeah. Before do an image, I fix the cell, so
okay.
That is not live imaging.
Okay.
And I spent, it depends, more or less like 20 minutes to produce an image in super resolution, with this technique. But the cell have to interact with the materials for, it depends, but for 7, 10, but also 20 days before differentiate.
Okay.
Okay.
Thank you.
Okay.
And this comes to my next question. So
okay.
You showed that you have the methods to see how this chromatin interacts with the scaffolds. What kind of applications do you foresee for this kind of method? Because I guess there are many.
Yeah. Now I'm doing my PhD and I'm working on the continuum of my thesis. And the next step is because the conclusion of my thesis is, okay, it's possible to go in super resolution with the cell culture on the scaffold, but now we want to know how change the chromatin of the cells on the scaffold. And so we are working with stem cells and we are working on the differentiation in osteogenic line. And this type of research I think are useful in the part of tissue engineering to understand what are the mechanisms that can induce the differentiation, and this could be useful also to modify the scaffold, the properties, and to find what are the properties that I need to differentiate my cells in a different lineage. So we are in the body step of the world of tissue engineering.
Okay. Any other questions? I think it's time. Okay. Thank you again.
Thank you.